In one section we get started.
Sure.
So I'll do a little introduction. Okay. Well, it's great to see everybody here. A lot of new faces that I haven't seen. I believe there's lots of friends. So, so today is our opportunity to celebrate the Flesher's PhD research and thesis. But it's a little bit of a bittersweet day for me, at least, because it means not only is he graduating, but he's going to be leaving us soon. And he's off as he he's starting a postdoc at Stanford. Yeah, a little bit of background. So I first met David at a Gordon Research Conference. These are meetings, and he went to this meeting as an undergrad, but they're not supposed to allow any undergrads. So somehow he snuck into this meeting, and I met him there and talked with him, and we visited and encouraged him to apply to Yale, which he did, and it was wonderful. He joined our group, and he's made such wonderful contributions. You're going to hear about his research where he's used cryo-electron microscopy to solve structures of a number of different proteins. But on top of that, he's been very much involved with the methods development, with helping us to develop methods to differentiate chlorophyll. He's going to tell us a little bit about that. But then he solved a number of important structures. He solved the very first structure of a photosystem with a site-directed mutation, a photosystem 2, and now I guess how many? 4 or 5 more structures of mutated proteins. So these are really opening up new opportunities for structure, for comparing the functional studies that have been done with the structures of these proteins. So that's really a significant contribution. And so in addition to all the wonderful research David has done, he's been, and fantastically engaged in the biophysics training grant. He organized the annual symposium. He's done a lot of work in that, hosts seminar speakers. He's done a lot more than just research. And he's really very much engaged. And he's been a wonderful member of our group, interacting with all of the others and all the collaborators that we've worked with as well. And so we're going to hear about that, and I'm really looking forward to the presentation. He's going to tell us 9 years in photosynthesis. And I told him he was going to make that the title of his thesis. Everyone's going to think it took you 9 years for PhD, and it didn't take 9 years. But I think that's when he started his undergraduate research. 9 years. So anyway, we'll hear about his 9 years of photosynthesis. So I'll turn the floor over to David.
Thank you, Gary. Thank you so much. I forgot to start voice recording, so give me 1 second. Okay, great. I'm going to actually continue on and tell a very funny anecdote of when I came right after the Gordon conference. Gary very generously said he was willing to meet with me on a weekend day in the morning. And I said, oh, that's great. I said, I'm in the area, but I was in Boston. And at the time, I thought Yale was in Boston. And so when I'm emailing him, I'm like, oh, yeah, it'll be easy. I'll just take a subway over there. And I put it out of my mind. And then the evening before, around 10 p.m., hey, Andrew, I look on Google Maps like, oh, what subway, what bus route do I need to take to get to Yale? And it zooms me out of the state. And I'm like, oh, no, what am I going to do? And I end up getting a Greyhound bus at about 5 a.m. in Boston, come to New Haven, meet with Gary, and then go back. And I told Gary this story at a Christmas party after joining his group, and he said, oh, I had no idea. And so, yeah, that was a fun story, and that was my introduction to Gary, and it was a good one. And so I want to thank you all for coming, and it really is encouraging to see so many people here today. And really, Gary had just the perfect word, is celebrate. Celebrate, my clicker not working. Okay, this thesis presentation is a celebration of really my story and my time here in New Haven. Like Gary was saying, I've been here for 5 years. The title of my talk is whimsical, 9 years in photosynthesis. I did 4 years in my undergrad, and I'll talk about that in a minute. But celebrating my science, celebrating the community that I've been a part of here in this past 5 years, and all in really good company. And so, again, I want to thank you all for coming here today and celebrating this with me. the rumor is that you're supposed to also learn a thing or two at these defenses. And so I'm also going to share some of the things that I've learned in these past 5 years in my PhD. Some about my science, some of the things that we've done in the group, some about what I think it means to do science and what it means to be a grad student, and then also some about life. So in other words, this talk is part science, part autobiography, part rambling of a geriatric grad student. And so I've organized it in these different chapters, again with that theme of trying to tell a story of my time here in New Haven. And I have these chapters, 1 through 7, and I'll go through them. The first 2 are science, and the last couple are community-based. And so, 9 years. What does that mean, David? You've only been here for 5 years. Well, I chose 9 years, one, because it's a fun title, and as I go into my postdoc, I'll be moving outside of photosynthesis. But I also want to do it in order to acknowledge and really celebrate a lot of the time that I spent in my undergrad. So I joined undergrad labs of Petra Froma, Kevin Redding, and Poland Chu back in 2016, and literally the first week of classes in my undergrad. And I spent 4 years, and I just had a fantastic time, really learned a lot, had fantastic mentors, some of which are in the Zoom room today, and I'm very happy about that. And so part of the talk, or part of the title, is acknowledging that as well. And I started working on cryo-EM and crystallography on photosystem 1 and photosystem 2 in my undergrad. And so also, glutton for punishment, you might say. In my PhD, I came out here to Yale. I also grew up in Arizona, if that wasn't obvious. Came out here to Yale and started my PhD, joined Gary's lab, also in photosystem 2. And so I've been in photosystem 2. I've been in this one protein complex for a while. And so that's also part of the romantic and whimsical 9 years title. And so I figure I wouldn't hold you in suspense, but Gary already mentioned it. My next step is I'm going back across the West Coast, long drive yet again, and I'm starting a postdoc in Stanford in the lab of Hua Chu, and I'll be doing cryo-ET and volume EM type work in different systems. And if you don't know who Hua Chu is, that's understandable. He's old, he's this guy. But for some fun, Richard Henderson's 2017 Chemistry Nobel Prize lecture, which was on cryo-EM, in the first slide, he acknowledges some of the people that were starting the field, and he names Hua Chu for organizing the first 3D EM Gordon conference all the way back in 1985. And yes, that means Hua is 78. And so that's where I'll be for the next couple of years. Oh, this is also a good time to preface. You'll notice there are a lot of cameras going on, a lot of Zoom. I want to acknowledge my parents who are Zooming in. They wish they could be here today, but they're doing the best and they're Zooming in from the hospital. And so I want you all to be conscious of that, both now and then also at the after party. I'm going to try to make it really accessible for them. All right. Chapter 1, Lessons on Nature's Water Oxidation Reaction. Okay, but first I thought you might humor me. Raise your hand if you know me from the department or from science. Put them up. Okay, great. Put your hands down. Raise your hand if you know me elsewhere in the community. Okay, that kind of speaks to really my life here in these past 5 years, and now there's a good mix of people, which I find encouraging. And also, I'm going to try to have a lot of interaction with the audience to keep you guys interested. All right. So, introducing this topic, 9 years in photosynthesis. What is photosynthesis? But my first question is, who here knows what type of flowers these are? Anyone?
Carnations.
Fantastic. Yeah, pink carnations. Bonus question, do you know, in the language of flowers, what pink carnations mean?
No.
Anyone? All right, I'll leave that for you guys to look up. But yeah, who thinks that pink carnations perform photosynthesis? I see some head nods, some hands raised. Okay, good. What about another flower? If this was a rose by another name would smell as sweet. Would it also do photosynthesis? Yeah, seeing more head nods. Name another flower that also does photosynthesis.
Dahlias.
Fantastic. Another one. Nobody knows any flowers.
Sunflowers.
Sunflowers. Okay, Fred is the only one who knows flowers in this audience. Okay. So we have this intuition that all these different types of flowers, these different types of plants are performing photosynthesis. But it's more than just plants. Here I have some moss that I got from my front yard this morning, and it's getting a little dirt everywhere. But I suspect you guys would nod along that it also performs photosynthesis. And if you have a little magnifying glass or microscope or something like that, this is a field piece. That's a 60 times magnification that you use to look at things in the field. If you looked, it almost looks like this tiny little microscopic plant. All these different ferns or plant structures. And that's true for moss, and also true if you zoom in more and more, things like algae or cyanobacteria. This is a rain catcher. We've had a lot of rain, but I dumped it out these past couple days. And so you can see there's green things growing in the bottom, things like algae and cyanobacteria. And so we have this intuition that they're all performing photosynthesis and that there's this huge genetic diversity and species diversity in plants, but that's also true in bacteria. Even though they're small and we can't see them, we think of them as all one type. But there's a lot of diversity. But even amongst all those diversity, they all perform photosynthesis, and they all perform photosynthesis in very similar ways. If you got a magnifying glass, really you need a microscope, much more powerful than this. You could get an image, something like this, with an electron microscope. And you can start to see some of the cellular structures that are inside these cells that are where photosynthesis is occurring. These are what are known as thylakoid membranes. And the reason that all these organisms do photosynthesis is because within these thylakoid membranes are these series of protein complexes here on the left that are known as the photosynthetic apparatus. And so these are the light reactions of photosynthesis, and they're what is being used to capture and store that initial light energy. So I'll pause and I want to ask the crowd again, what is a protein? What do people think of when they think of protein? Quiet crowd. Any suggestions?
Muscle.
Muscle. Great answer. Anything else? I think of steak. And this is a steak I cooked up. And really, what's inside this steak is proteins. And you say the word protein, most people think of it as the food. That's just this general class of all of this molecular biological thing. And so proteins are formed by amino acids. Anyone here use Legos? Everyone here knows what Legos are. You can think of amino acids as different pieces of Legos. And so maybe one amino acid is this blocky 2x2 piece. Another amino acid is this long, thin piece. And by combining all these different pieces, you can form different, what are known as secondary structures, tertiary structures, quaternary structures, form these different structures. Here are some examples of what those Lego pieces might look like. In biology, there are 20 that are naturally occurring. And together they combine in these different sequences and these different structures. And really they give this huge variety, huge diversity in structures of what proteins are, what their shape is, how big they are, what they do, all these sorts of things. So even though when we think of protein, I think of steak, really you should be thinking of them as these tiny little nanomachines. And some of them are really simple. Some of them are really simple tools, something like a hammer, or maybe a screwdriver, one a Phillips, one a flathead, that sort of thing. Some larger protein complexes, things like enzymes, are really complex and really delicate, something like a Swiss watch. And if you crack open the Swiss watch, you can see that there's a lot of different pieces involved, and each of these pieces are really important. And so that's sort of a theme of my research. I'm looking at protein that is really complicated, really complex, and has a lot of these finer details. And really what I'm trying to do is crack open this watch, look inside, fiddle with it, things like, hey, this gear has 40 teeth. What happens to the watch if we change the gear to have 30 teeth? What if I changed it from 40 to 50? Does the watch still tell time? Does it run fast? These are some of the intuitions or sort of ideas that you could think about when thinking of proteins, and something I've thought about during my PhD. And so keep that analogy in mind. Nano machines, little tiny machines, and also the Swiss watch in changing things. And so back to this view, all these different protein complexes, even though in this diagram they're just circles, are these very complex little machines. And then we need to try to understand how these machines perform their function. All right, here I'm showing and in our lab we're particularly interested in photosystem 2, abbreviated PS2, shown down here on the left. Because of its ability for this machine, it oxidizes water. All right, that's really hard chemistry to do. I'll talk more about that in a moment. And so here on the right is what photosystem 2 looks like. For the experts in the crowd, it's this membrane protein, very large membrane protein, 800 kilodaltons, 21 unique subunits, C2 symmetry. It's about 30% cofactor, which is relatively rare for proteins. That's because of all the chlorophyll pigments that are shown in this transmembrane region. Those are the pigments that are performing that light capture. They're absorbing the color, all right? And so this is the big nanomachine that I've spent a lot of my PhD looking at. And also, if you went to elementary school in the 20th century, I guess, or 21st century, you might have seen a diagram like this, and this sort of simplification of what a photosystem is. And so photosystems will capture light energy in this antenna and transfer it and do some kind of photochemistry. And so really what photosystem 2 is doing is it's doing water oxidation. It's doing this reaction down here on the bottom left, where it's taking electrons and protons from water molecules and releases oxygen as a byproduct. And I'm sure we all know that plants produce oxygen, but it turns out that that oxygen is actually coming from water molecules. Who here has also heard the phrase, the mitochondria is the powerhouse of the cell? Yeah, seeing head nods, hands raised, good, you're learning. Well, photosynthesis is very similar, and these thylakoid membranes and chloroplasts are really analogous. They're the powerhouse of the plant cell or the algal cell. And the reason is largely because of photosystem 2 and other protein complexes that are capturing and storing light energy. And so photosystem 2 uses water and light to fuel energy production, making it an attractive system to study and emulate for green energy. So that's been a lot of the impetus for my research, is trying to understand that reaction more. All right, I'm going to be taking it a step up a notch here. And so the question, how does photosystem 2 perform water oxidation? There's this cofactor, this OEC cofactor, metal cluster, that's embedded in the protein complex. And if we zoom into that, we can see that it's embedded in this really highly conserved protein environment. And it's this cluster of 4 manganese cations and a calcium cation. And it's surrounded by these water channels. These are water channels that are cavities within the protein that are delivering the substrate, in this case water, to the active site for water oxidation. And the reaction cycle is shown here on the left, what's known as the Koch cycle, where this metallo cofactor becomes gradually increasingly oxidized. Electrons and protons are abstracted at different steps, and then ultimately OO bond formation occurs, oxygen is released. That's the stuff that we like, but for the plants and for photosynthetic organisms, it's just a byproduct. And then the catalyst is refreshed. And so that cycle continues, and this is how energy is being stored. Who here likes money? Maybe that's a bit of a trick question. But if you ask the plant, it doesn't actually care that much about money, even USD. What it cares about are electrons and oxidizing, reducing potential. And so really what's being stored is currency, cellular currency, biological currency in these electrons. They're not actually just free-floating electrons, they're reduced equivalents, but I won't get into that. And so we have this question. We want to ask this question, how does the OEC oxidize water? We have this general reaction cycle, but a lot of the molecular details are still unknown, even to this day, after decades of research. But when you ask this question, how does the OEC and in photosystem 2, how does this nanomachine oxidize water? How does it store the energy? There are these subtler questions that are involved. Things like, how is proton egress controlled? In these different steps, you see an H+, where does that go? How is that managed within this protein environment, within this nanomachine? What does the electronics, or how does the electronic structure impact the mechanism? To the experts in the field, or in the audience, I said the OEC has become gradually increased in oxidation. How does that affect the structure and the energetics of this tau cluster? Which side chain chemistry is essential for which steps? If you have this protein environment, these different Lego pieces, these different amino acid Legos, which are really important for which steps, and which ones can you change and maybe not affect things too much? So these are sort of subtler questions that we've tried to answer and address on this broader goal or theme of how does the OEC oxidize water. And so how might you sort of get at this question? You can use site-directed immunogenesis alongside biochemical and biophysical methods, study the active site of photosystem 2. That's been done. What that means is essentially you're changing out one amino acid for another, and you're trying to understand the chemistry, the chemical consequence of that. So you're changing your Lego piece from a 2x2 block to a really long one, or something like that. You can think of it in that way. And then in recent years, the Brudvik lab has started to structurally characterize some of the most interesting site-directed mutations using CryoYam. And so we have these series of papers. I'm going to just talk about one of them today, try to keep things light, keep things brief. First, I figured I should give a general scheme of what these experiments are, what we're doing. This is a general experimental design for single particle CryoYam. Is what I'm doing. And so what we do is we take our cells, in our case cyanobacterial cells, but a photosynthetic cell, and long story short, we purify the protein. In fact, I say we, our great collaborator Rick Davis, who's on, does a lot of this and is really excellent at it. And so we purify our protein, and then we can apply it to what's called a CryoYam grid. It's this tiny little disc, thin as paper, only about 3 millimeters across, hard to even see. And you put 3 microliters, less than a droplet, on, and then you plunge it into liquid nitrogen, you freeze it, image it on an electron microscope. That's something, a lot more powerful than this, but when you're looking at these small details, you need a very powerful microscope, and this one actually uses electrons, not light. And you get images, you do data processing, define the orientation of your particle on these images, and then you gain CryoYam density, and then you can model your structure into that. All right. You just rambled on, David. What does that mean? Well, it's a little bit like this little GIF that I have. There's this person who has this Lego structure, nanomachine, here, and it's casting a shadow, this light that's being cast upon it. It's a little bit like what we see on the micrograph, on the electron microscope. And so it's casting these different shadows, and that's sort of what we see, the different orientations of the protein. And as you rotate this object, if you get these different views, if you get enough, and if you're smart enough, or really the software is smart enough, you can get a reconstruction of exactly what this Lego block looked like. That's sort of the intuition that you're dealing with. You have a shadow on the wall, and you're trying to then trace back to what is the exact structure of your Lego block, or in our case, the photosystem. All right, going ahead, I'm going to talk about one project, this V185N mutant structure from organism Acinetocystis species PCC 6803. Already in that, I'm starting to just plow ahead and leave some people behind, so just hold on tight. But speaking to some of the experts in the audience, this is a conversion of a valine amino acid side chain to an asparagine. And so we're changing the chemistry from a nonpolar group to this group that's able to be involved in hydrogen bonding interactions. And we solved this to a very nice high resolution of 1.99 angstrom, global resolution. And the reason why we're particularly interested in this mutation, like I said, there have been these mutagenesis studies looking and trying to understand the active site of photosystem 2 more. This particular mutant has several really interesting, there have been really interesting studies on this. So it has about a 40% steady state oxygen evolution rate relative to wild type. So what that means is it is still performing water oxidation. This protein still functions, but it functions at a lower rate. It's worse. It's worse off for this mutation, and it's actually quite a significant effect. probably the most interesting observation, it has delayed O2 release kinetics. It goes from 1.2 millisecond to 27 milliseconds for that time scale. And so what that is, if you go back to this diagram that I showed before, this S3 to S0 transition, S4 is this transient intermediate. In this step, oxygen is released. And you can measure the time constant that oxygen is released. And that tells you some about how that OO bond formation takes place. And that's really the interesting chemistry that we're trying to understand more. How does OO bond formation take place? And these results were interpreted to result or attributed to an increased entropic barrier, particularly thinking about hydrogen bond transfer or proton transfer. I'll come back to that later. Some other interesting observations were it has altered substrate water exchange kinetics. These are kinetic isotope experiments done by another lab looking at trying to find out what are the exact oxygens that go and become molecular oxygen that's released. So is it a mu-oxo? Is it a water molecule? Looking at those questions. And then also this general location of V185 in this protein environment that I have highlighted here on the right, this is really what people think about as the active site of OO bond formation. They don't know exactly what's going on. They don't know which are the exact substrates. There are some prevailing theories, but this is the general region where OO bond formation is taking place. And so a mutation at this site is inherently sort of interesting in how it might be affecting OO bond formation. Really, in the field, people talk about two conformations. That means sort of how does this amino acid orient itself. One where it's facing this chloride 1, sort of swinging down right here. Another one where it swings up and interfaces with the OEC, interacting with the OEC and maybe some waters. And in literature, this OEC facing conformation is favored. It's what everyone talks about, and it's what my hypothesis was going to be that I was going to see. But going into this question, we had this, going into this experiment, we had this question, can we identify the correct V185N side chain conformation and correlate our observations to the previous biophysical data? Can we learn some things about water oxidation through this mutant? And so, long story short, we solved a very high resolution structure here. People in the field know that you kind of want to show off here and show some of the quality of your features. For people who aren't in structural biology fields, what I'm showing is the sticks or the circles are sort of the atoms that we model based on experimental data. The data itself is shown as the surface or the mesh. And so you fit your atoms within that experimental data, within the mesh, and that tells you things about the protein structure. And there's another quote, structure determines function in biology, and so that's what we're trying to understand. And also, if you're not familiar, this is extremely high resolution. I actually meant to look this up, but very few deposited structures are better than 2 angstroms. And so at this rate, we have a fairly large percentage of all the structures that have ever been determined at this resolution. I might be getting up to the double digits, to be honest, percent-wise. So, okay, so that's what the experimental data is, and I'm going to be showing a lot of plots like this. All right, jumping right into it again, we see, looking at this V185 position, what do we actually see in the data? What is the actual conformation of the V185N side chain? Well, it's not the OEC-facing conformation, which was the prevailing hypothesis in literature. Instead, it's definitely, certainly 100% this chloride 1 facing conformation. You can see that in the density here in the surface, and also this isomesh at a different contour level. And so the V185N side chain adopts the chloride 1 facing conformation. There's no evidence for multiple conformations. And so that was already a very exciting observation and conclusion to make. And it makes us now have to rethink some of those previous biophysical data that were interpreted using an OEC-facing conformation. Of course, that now prompts another question. If you're an expert in cryo-EM, you'll know that you can't really differentiate a nitrogen atom from an oxygen atom. Nitrogens are blue, oxygens are red. And this amide, is it oriented like this, or does it, which spin? And so in cryo-EM, you can't differentiate these different atoms, but that's a really important question to answer when we were trying to think of the chemistry in this active site. How does this mutation affect the active site? And so there's a question, what evidence can we gain for a precise asparagine 185 conformation? And actually, we can use secondary observations and get pretty confident. What I'm showing here is sort of a zoom out of the active site, emphasizing this chloride 1. This is a chloride ion or a chloride cofactor that's conserved and very important for the active site. And it's also in this chloride 1 water channel. This emphasizes the chloride 1 coordination sphere. What are the amino acids? What are these Lego pieces that are confining and keeping this chloride 1 in the structure? And it's very highly coordinated. And so we can do this comparison where we can see V185N, that's the mutation we made, and then it's also this wild type, this conserved natural asparagine side chain that also is a ligand coordinates chloride 1. And so if we look at how N181, coordinates chloride 1, we can see that it has a conformation like this, where the mean of the amide points down and has a bond geometry where the hydrogen is pointing directly at chloride 1. And so it's providing a hydrogen bond interaction to chloride 1. Chloride 1 is negative. It wants positive charges. It's nucleophilic. And so if we then compare this sort of bond geometry to what we see in V185N, if we orient the amide like this with the mean pointing up, we can see again we have a hydrogen bond that's sort of pointing down towards chloride 1. Here I'm showing the hydrogens, even though they're not modeled in the experimental data. Hydrogens are fairly predictable, but they're just shown to emphasize the bond geometry. And so the bond geometry is not as good as N181, but it's still quite excellent. The distances are also very reasonable. And if you sort of imagine, if instead this amine was pointing down where this red oxygen is, these hydrogens would just be pointing out sort of into empty space. And so they wouldn't be able to provide a hydrogen bond to chloride 1 and stabilize that. And so based on this observation, we have a pretty convincing conclusion that the V185N side chain is oriented like this, with the amine pointing up. And that's also important because that means it provides an additional hydrogen bond facing upwards, maybe forming transient interactions with water 2 and water 61, these conserved waters, though the bond distances and bond geometry are quite poor for that. All right, so that's some of the observations we can make in this structure. There also appears to be a weaker lysine 317 chloride 1 interaction leading to an alternate lysine 317 conformation. What does that mean? So I have this diagram here on the bottom left and sort of hinting to that clue. If you go to this other perspective, you can see that lysine, which is this other conserved side chain that's in this chloride 1 channel, and very prominent in this chloride 1 channel, it has this orientation where it's facing this chloride 1. That's sort of the wild type canonical orientation. What we see in the data is we see this alternate lysine conformation, in which the lysine is sort of swinging away, and there's reasonably strong evidence for that in the experimental data. And if we look sort of more broadly, what does this mean for the chloride 1 channel to have this alternate lysine conformation? Well, it's swinging down and it's putting it within range to interact with this conserved glutamic acid side chain, this E312. And so if E312 can interact with the lysine, that might change the conformation of E312 to swing up, have this alternate conformation, and then that might also induce another conformation of E65. And it's changing how this hydrogen bonding network in this chloride 1 channel is posed or oriented. Now, we don't have very strong experimental evidence or cryo-EM density for these glutamic acids. Carboxylic groups, they don't show very well in cryo-EM data. I won't get into it too much, but this model is actually really consistent with some recent models for proton transfer through this chloride 1 channel, where this quote-unquote closed conformation of E65 and E312 interacting with each other, if E312 is induced somehow to change its orientation, interact like this, E65 might also change, and then a proton could be transferred through this quote-unquote gate. And at the time, that was just suggested, but this is some really interesting evidence for this lysine that might be causing this conformational change. We can also think about the chemistry a little bit. of why would we observe this alternate conformation if this lysine is sort of waiting and right at that threshold to want to change this conformation and weaken its interaction with chloride 1. If V185N is donating this additional hydrogen bond to chloride 1 being nucleophilic, that would weaken its interaction with the lysine side chain and maybe pushing it into this alternate conformation equilibrium. So that's a really, really interesting observation we were able to make in this data, and it was a surprise, and so that's always great. There is some modest evidence for perturbed hydrogen bonding network and shifted water molecules in the chloride 1 channel. I won't go into that for the sake of time, but I can answer questions if anyone's really interested about it. All right, so David, you just rambled on quite a bit. I see a lot of people being like, what's he talking about? I went through, and so that's work that's been in the paper. What am I really saying? Who here recognizes this image? I see some hands raised. I had this. This is a fridge magnet toy that kids will have, and it's a marble run. So you can put a little marble on the top, and you can rearrange all these different pieces and have a marble kind of go down and funnel through and end up in this bucket. It's a really fun toy for kids, actually. I'm honestly a little surprised that more of you haven't seen this. So it kind of ruins my analogy. But another view of this is this diagram is you have this channel or this section of all these different pieces, and they're all positioned in a way to support this marble kind of funneling down through. And that's actually really similar to what we're thinking about in this data and what I was just spending a lot of time talking about. We have this chloride 1 channel, and really what we're thinking about is how does this channel facilitate, how does it help control proton egress? Because proton egress has to be controlled when thinking about entropic barriers for O bond formation. And usually when we think about this through a Grotus type mechanism, where a proton sort of bounces around through this Grotus competent side chains and water molecules. And so it's really similar to this toy, actually, where a water molecule is kind of funneling down, and these might be important side chains that must be conserved to facilitate this proton transfer. And so maybe this lysine is analogous to this little guy right here that sort of catches the marble and then swings it down. Catches the marble, swings it down. If this was in the wrong conformation, wrong position to accept that marble, it could slow things down. And so that's the analogy to try to understand a bit of what I've talked about. I was quite happy when I came up with that analogy, but I guess most of you guys didn't play with that toy growing up. All right, so in conclusion, what am I saying about this V185N structure? We went into this experiment. Can we identify the correct V185N sidechain conformation and correlate our observations to previous biophysical data? We were able to solve this very high resolution structure, which is great. It's important that we get high resolution so that we can see the water molecules and make observations of the protein structure. We see that V185N clearly adopts a chloride 1 facing conformation and donates a hydrogen bond to chloride 1. That's a very important observation and is caused people to have to reinterpret some previous results. We see that this lysine 317 side chain adopts this alternate conformation, and this might be consistent with recent models for proton transfer and how proton transfer can be affected in the chloride 1 channel. And then, like I said, there's modest evidence for perturbed hydrogen bonding network in this tiny little diagram, but I won't get into it. All right. That's one vignette of one of the projects that went into my thesis. So gave you a little bit of a taste of the type of research that I've done. For the sake of time, I can't go over all the research that I've done, but here I have sort of a summary. I'm going to walk you guys through a brief few words and also just kind of show some of the work that I've been a part of. So including this last paper, what I just talked about, which is in review, and we've received favorable reviews, and so hopefully we'll get that submitted soon, and it'll be accepted, and that'll be great. I've been a part of 14 projects during my time here at Yale in these past 5 years, and there's a quote: "Make hay while the sun is shining," and it's really been great. Of these, 3 have been first author, 3 have been co-first author. Some of my amazing co-authors are in the room or on Zoom, and I'll talk about those in a moment. 3 papers have been on these different protein structures. I just talked about this one here on this V185N mutation. I could give similarly confusing presentations on other mutations shown in these diagrams. I also have this whole series of papers that I collaborated on that Gary mentioned in the introduction about forward light photosynthesis. For the sake of time, I'm not going to talk about any of those or go into that detail, but essentially amongst these 6 papers, we solved structures, protein structures. We did analysis, developing methods. Jimin has this great cone scan, and we expanded on that idea. And so these are just some figures sort of representing those works. There's also this really kind of funky glycerol paper. It was a one-off. This is a fun paper that was kind of something that was left by a previous student, and Jin Chan and I collaborated and fixed that up, or finished that up. And it's this kind of interesting study showing how delicate the hydrogen bond network is in photosystem 2 and how it can affect the energetics of the OEC and electronic structure of the OEC as detected by advanced spectroscopy, EPR spectroscopy. I've also, anyone in science knows that you get on papers where you didn't actually do that much. And so I've consulted, generated minor data on several other papers, and those have also been great. And these are actually really important. It's really important for you to bounce ideas off people in your lab. And it strengthens your own thinking, strengthens their thinking, and is an important part of science. And being generous, I also think, is an important part of science. Not to discount myself too much, though. And then also, this is a funky paper on that topic. This is one from a rotation I did in my very first year in Jun Lu's lab, which is a tomography lab. Really, all I did for this paper was generate the data for this figure. But it's a bit of a foretaste of what's to come, because going into my postdoc, I'm going to be doing a lot more of this type of research, tomography and volume EM. All right, now I'm getting into the more retrospective part of my talk. And so I have a couple quotes as I'm going to walk through, and photos. There's this quote by, oh, no, I'm supposed to ask you guys a question. Okay. Raise your hand if you are in a PhD program or some kind of professional program. Professional school, master's. Stick them up. Okay. Keep your hand raised if you're in the first half of your program. Okay, quite a few people. You count. Yeah. Okay. And so, in addition to talking and sharing some of my views and some of the things I've learned about my science and the topic, I figured I could also share some of my experiences about science and how we do science, and speak to you. And so there's this quote. It's a quote by a postdoc in the lab. He just said this for fun, but it's poignant. Getting a PhD isn't supposed to be easy. And that's true. It's really hard. It's not just technically challenging. Doing the research is technically challenging. Thinking about writing, thinking about conclusions is difficult. But it could also be really emotionally challenging. It can be tough to have tenacity. And sometimes an analogy is it's sort of like you're staring at a watch face that doesn't have a second hand. And you're trying to be really diligent and be on top of looking at this clock. And as hard as you look, it doesn't look like it's moving at all. And so that can be really discouraging for a lot of students. And throughout your PhD, almost everyone will go through this season of discouragement. But I think it's really important to have the perspective to take a longer view on things. A lot of us go into PhD programs immediately out of undergrad, something like that, where periodicity, a semester system, really emphasizes that things happen in these week basis or month-to-month basis. In a PhD, that can be different for a lot of people. And so I want to encourage everyone to take a longer view on things. So only when you maybe look away from the clock and look back, you can see, oh, wow, the time has changed quite a bit over the years. And so try to give some encouragement and perspective there. There's also an attempted quote to remember line by James Choi. Some of you know who James Choi is. Your goal should be to work faithfully, not fruitfully. Again, it's not a direct quote, but what does that mean? It means that sometimes you're not really in full control of your results. Sometimes experiments don't work, and sometimes it can be really frustrating. Even though you put a lot of effort into something, it doesn't pan out. And sometimes you put a lot of effort into something and things go really quick. And so don't get so attached in satisfaction of exactly the results that are coming out. Instead, try to just focus and work faithfully, show up every day, try to do a little bit better, and the details will work themselves out. And then also a quote that's maybe by Teddy Roosevelt, comparison is the thief of joy, which I think is a great quote, and just try not to get discouraged. And so that's, I shared some about science, some things I think about science. Now I'm going to get into other parts of my talk and give some science acknowledgements. First and foremost, I really want to acknowledge and thank my advisor, Gary Brudvig. He's a fantastic advisor. I printed out my thesis book. It's over there on that table. And I'd love for everyone to sign it or write a little note. It's open on a page. And at the very beginning of my thesis, and all theses, there's acknowledgements, and I have this line that I thought I'd share. I thank my advisor, Gary Brudvig, for his unequaled guidance and mentorship throughout my PhD. Truly, I have never met a more admirable faculty in his wisdom, kindness, and professionalism. And it's certainly true. I've learned a lot from Gary, both science, both how to do science, how to be faculty, or how to engage with the department. And really, I couldn't have gotten luckier with an advisor. So thank you. Also, if you're a nerd, and this is leading into the whimsical side of things, about the Lord of the Rings. In the Lord of the Rings universe, elves are these really wise creatures, and so that's some context of this quote. But this is a great quote that I read it. I'm like, oh, that's Gary. So this is Frodo seeking advice from the elf Gildor when fleeing the Shire in the books. So this is Gildor speaking. But it said, "Do not meddle in the affairs of wizards, for they are subtle and quick to anger. The choice is yours to go or wait." And Frodo says, and it is also said, answered Frodo, "Go not to the elves for counsel, for they will answer both no and yes." And Gildor laughs. "Indeed, is it indeed," laughed Gildor. "Elves seldom give unguarded advice, for advice is a dangerous gift, even from the wise to the wise, and all courses may run ill." And if you know Gary, you know that sometimes he can be a bit of a sphinx, and it really highlights his wisdom. It really highlights his wisdom. And it's something I really appreciate from Gary. If you know Gary, you'll like that quote even more. All right, I also want to thank a few other people. My friend Chris Gisrael, a postdoc who was in the lab. He's no longer with us, unfortunately. This is an inside joke. He's not with us because he started an independent position at Wisconsin-Madison. I'm hoping he's on the Zoom and found that a bit funny. He's been fantastic. We've had 11 papers together. I've actually known Chris from my undergrad at ASU. And so we've known each other for, I guess, about a decade at this point. He's fantastic. I can't speak higher about him. I also really want to thank Rick Debus, who's been a fantastic collaborator. We've had 5 papers together, and has really been instrumental in a lot of this mutagenesis work. And he's my outside reader, actually, on my thesis, and so he's zooming in. I also want to thank Jin Chan, my classmate. We came in the same cohort, same as Matt, and we've collaborated on a lot of papers, really engaged together, and we have a collaborative group, and so in a lot of ways, Jin Chan feels like a member of our lab, and in a lot of ways she is. And we've had 6 papers together, and so I really want to also acknowledge Jin Chan. And then more than that, we have this, like I was saying, this big collaborative group that is great. We discuss science. It makes our science sharper. We ask questions that we don't know. We see different perspectives, different types of projects. And we meet together every Friday over Zoom. And so we meet with people here at Yale, like Jamin, like Victor, and then also people from other universities, PIs from other universities and their labs. And we get together every once in a while and fellowship together, and it's great. I also really want to thank the Brudvik Lab. The Brudvik Lab is on 2 different campuses, and we get together and we do events. And so these are some of the figures. Most recently, a couple of us got together and ran the New Haven Road Race. Gary was a beast and ran the 20K. I ran the 5K. And we had a great sign, Go Brudvik Lab. And so that's great. We've also just had great meals together. Here we're celebrating Gary getting into the National Academy this past year. We do these things where we'll have meals. People will make a meal about their sort of based on where they grew up. So this was Italian dinner because we have some Italians in the group. Gary's also been very generous, has invited us up several years to New Hampshire during the winter, and we've gone cross-country skiing together. We've shared meals. If you don't know, I like to cook, and so I like to make a lot of meals, and so this is showing this. This is a ratatouille I made. This past year, or recently, we also went and saw the solar eclipse, the total solar eclipse up in Vermont, and here's a great photo of that. Big smile. And here's a dinner that I had. I made a bunch of Vietnamese food, some egg rolls, the cursed egg rolls. I've told some of you that story. And we made spring rolls. And again, we've had great dinners. And so really fantastic group. We've also gone to the OEC. And this is an old photo, but we also went and saw a baseball game and even had someone's birthday celebration up on the slides. All right, chapter 3, moving to New Haven. I probably should have told you guys earlier, you can interrupt me and ask questions if anyone has questions. Maybe I should pause and say, if there are any scientific questions, let's hear those now, because the rest of the talk doesn't have anything to do with science. Speak now or forever hold your peace. Guess I'm getting a PhD easy. Nikhil? Nikhil's on my committee. I guess I'll point out, my committee members, Gary Brudvik, Nikhil, and Yong. And also, Rick is an outside reader.
Amazing work, David. Congratulations.
Thank you.
So this is one failure I'm really envy that there is this huge amount of biochemical, biophysical data, and you should give examples where how your structure agreed with the prior data, but did your structure find something contradicted prior data?
Oh, yeah, in this last work, well, okay, so that's sort of a philosophical question as well. People have said, I had an old advisor back at ASU that said the data is never wrong, just your interpretation. And so I'll say that some of the interpretation has been wrong, especially like I was saying about this V185N sidechain, where people are interpreting it on the basis of it's interacting with the OEC, and particularly thinking about substrate water exchange and how it affects proton transfer. Whereas in the structure, we definitely see that it does not interact with OEC. It's interacting lower in the channel, and so it seems like these effects that people have measured, substrate water exchange, kinetics, things like that, are a result of its effect further down the channel, not directly in that first ligand sphere, directly interacting with the OEC or substrate waters. And so, yes, we have. I told you guys the coolest story, though, so there's also that perspective. We've also seen some things where in the structure, we thought a lot of things were going to change. We crack open the structure, things look pretty much all the same. And so that's also the other side of things. Yep.
When you're cooking, what's the state of PS2? Which parts of the reactions? How do you get it?
That's a great question. There's a lot of, so photosystem 2, in the field, people have measured, it's turned over very frequently, a time constant of about 30 minutes, if I'm remembering, because it gets damaged so frequently as it's creating reactive oxygen species. And so in cells, a lot of energy goes towards disassembly, reassembly, and repair of photosystem 2. And so you'll get different conformations. See you, Nikhil. We purify either biochemically, but certainly in silico purification of the full intact photosystem 2. And if your question is about that reaction cycle, there's a dark stable state, the S1 state. We try to put all our structures in that. but that's a really interesting question because there is XFEL studies, X-ray free electron laser crystallography studies that try to look at the structure in higher S states. So as you go through their reaction cycle. That's not what my work has been on. My work has been on a static perspective, which is a limitation of our studies, actually. So, no?
This is actually not a question, it's a comment, actually, especially to the audience not from Yale. So David showed a list of 14 publications. Just want to comment actually for probably above average PhD at Yale. It's about 3 to 4 papers. So this is extraordinary.
Thanks, Joe. Any other questions? Speak now or forever hold your peace. I'm going a little bit long, but too bad. All right. Any questions on Zoom? Chris says, I am alive. Good. All right, chapter 3, moving to New Haven. So some of you know I moved here from Arizona, long trip. And that first fall, I thought, oh, very pretty. And I want to get this on record very early, me complaining about yard work, because I hate it. The leaves were very pretty until I learned I had to pick all this up, in which I was not satisfied. And every fall, I'm annoyed. And fortunately, it seems like I'm going to be gone before I have to pick up the leaves again. Also, that first year, recent years, the winters have been pretty mild. That first year was kind of snowy. We had some big snows. Here's me. And again, I was not impressed with shoveling my driveway. That's my mailbox, and I had snow up to the top of my mailbox. I also have a very long driveway, and it's silly. Oh, if you guys don't know, as tradition, you spend some time, so you talk about science, then you spend some time going through a photo album and sharing moments. This is, again, in efforts of celebrating my time in New Haven, moments and also communities, which I'll get to. I've also mentioned to quite a few people that I moved into this house and renovated a bunch of it, and also packed up this U-Haul. Have way too much stuff. I can see things in this photo that I moved here and didn't touch for 5 years. So, yikes. But here is my dad and I. I'm not in any of these photos. Scraping wallpaper off the walls and refinishing it. And if you've been to my house since then, hopefully you think it's quite nice, and so I'm happy that worked out. Here's some other photos of me living the American dream, mowing my lawn, drinking this is soda, not beer. I'm very sad that this lawnmower broke, and now I'm slumming it on. I have a push mower. And here I am with my dog, Chewy. Here's my sister and my brother. We're changing a flat tire when we should be going to an event in New York. And then also, finally, I really want to acknowledge my parents and celebrate some of the times that they've come out to see me, going little walks, schooling them in board games. Here we're playing Terraforming Mars. And then also, I always rope them into doing a house project when they visit. And here we are building a retaining wall in my backyard. There's a lot of work, but has been good. And so, again, really want to celebrate my parents and shout them out as they're listening in over Zoom. I also want to shout out some of my friends. And if you know about me, I don't take very many photos of myself. My entire 4 years of my undergrad, I didn't have a single photo of me in the lab. Turns out, I don't have a single photo of me in the lab still, which, oops. But these have been my best friends since high school, Bobby and Tamara, and this is the first photo. I can't find any photos of us. Here's one back in prom. And it's been a couple years since then. But yeah, they've been really encouraging, really fantastic friends, and we play a lot of video games together. And so we keep in touch remotely. They were in Arizona, now they're in Omaha, and we keep in touch. Also want to shout out Andrew, who drove all the way here from near Boston. He's also been a really fantastic friend these past years. We've known each other for, somewhere between 10 and 15 years now. Yeah, a while. And so, yeah, we've kept in touch, kept good friends, and yeah, friends are encouraging. Okay, so on the topic of moving to New Haven, I spent a lot of time during the move thinking. It's a long drive from Arizona to Connecticut, and I'm also retrospective because in a lot of ways, I'm doing the same thing again. I'm moving across the coast to a place where I don't know anyone, and thanks, Kevin. I saw your message. And so I want to ask the audience in this contemplative moment. There's a quote by Warren Buffett that I like. When you get to be my age, whatever reputation you have, it's probably the one you deserve. And as a geriatric PhD, I think about that. And so this is a rhetorical question, but I want everyone to think of an answer. Actually think of an answer in your heart. What do you want to be remembered for? That could be broader than just your entire, or it doesn't have to be as broad as your entire life. A lot of us are in PhD programs and have this, I think, incorrect periodic view of their life, that they're just here to finish their degree, just get a program, and then they're going to leave and start the rest of their life. But that's not true. I actually talk about that more later. So what do you want to be remembered for? Well, I can't speak to what my reputation is, and it's maybe different amongst different people. But I'll share some quotes that speak to me and type of life I try to live. This is a fun one by Winnie the Pooh. This quote doesn't have anything to do with Winnie the Pooh, actually, but your greatness is not what you have, but what you give. And serving others enriches the giver's soul, paraphrase. And everyone to whom much was given, of him much will be required. And so I think of these and try to have had an impact beyond just what gets published, what I do in the lab in these past 5 years. And I think part of that speaks to the community and who's shown up to my defense. And then also some funnier quotes. Well, the years start coming and they don't stop coming. Smash Mouth. And that's true. That perspective that you're only here for your PhD and then you're going to be done with it, that's not true. Your to-do list, so to speak, is never going to end. You're always going to have things in the way. Things that, I need to do this. I don't have time to do that. And there's other quote from this movie. I won't read all of it, but I'll just say there's this mention. Virtue is not convenient at the time. This will not suffice. You're always going to have reasons not to do things, to be too busy to contribute. And I think it's still expected of you to find the time and to serve. Okay. I've tried to do some of that in the department. Like we were saying, I've organized some symposia and organized student speakers. I was the GSA rep, though I didn't really do anything for that. Organized summer barbecues. I had to do all the MB&B department ones the summer of 2022. COVID was a little bit weird for our cohort. But launching in, I've also tried to do that broader in the community. And so here, I'm combining chapters 4, 5, and 6, Mondays, Tuesdays, Thursdays, Fridays, and Sundays in general. These are all fellowships that I've been a part of, Christian fellowships I've been a part of. And so most of my weekdays are actually pretty full. I'll go to a Bible study. If you don't know what a Bible study is, it's sort of like a book club / support group. And it's about 2.5 hours, 3 hours. And Mondays, I've been part of the Yale Graduate Student Christian Fellowship, YGSCF, which has a horrendous acronym, but that's what we're stuck with. This past little while, I've been a leader of this group, and so and want to celebrate how great and enriching that has been as well, and share some fun photos, and also see some familiar faces in the crowd. Here we are making ice cream, some soft serve ice cream, and here we are watching a movie night. We'll also do socials every so often, so most of the photos come from that, actually. It's not the most active photos of us just sitting around in a circle talking. Here is this really fantastic Christmas party that Renee and Daniel put up. A very cute photo. And then also this tradition where they bake a bread and there's a little baby inside and a little porcelain thingy. And whoever gets the slice has to wear a hat. And so Renee ended up having that slice, and so she's got a little crown. And then also someone has to sit under the table. Also going to see some or listen to some singing, having different dinners, Thanksgiving dinners, going on hikes, seen Amelie speak at the International Church. Yeah, other hikes, other parties. This is a Ren fair, and so yeah, really want to celebrate and record some of my involvement in that group. Photos keep coming. This is actually some older photos of some people that have come and gone, but are still remembered. And then this is a matcha milk crepe cake that I made. Turned out not to be very tasty, but looks really cool. And then also there's a funny story for people who know I did another milk crepe cake that spilled on the floor. And it got too tall. Anyways, all right, so those have been Monday nights. Then on Friday nights, I've done Rivendell. That's actually where we're going to go right after this. And so Rivendell is again, a group and show a fellowship of different people, different professional schools across the different campuses, engage with different people. And here we are meeting, having a little Thanksgiving. You can see Taylor back here cutting up the turkey. You can usually catch me on Friday nights in the back room doing dishes. Here I am at the graduate student fair a few years back, welcoming new students. We'll go on different retreats. I've had people over for barbecues, and so that's been great. I wish I had more photos I could share. And then also, I'll say Tuesdays, Thursdays, Sundays, general involvement. These are Bible studies affiliated specifically with Trinity Baptist, which some people in the crowd from. Long story short, I was going on Thursdays, switched to Tuesdays. Won't get into it. Here we are getting together for some food. Point out, this was mine. It's this shepherd pie. It turned out great. And then, yeah, getting together. I had this big barbecue, and we got together, played some board games. If you don't know, I love board games. So there are quite a few board game pictures. In fact, for Trinity, recently I've put on a couple of board game events as a social and encouraging other students or the congregation. This is 99% all my board games. So I have a pretty big board game collection. That's now boxed up, sadly. But yeah, all huddled in the basement, playing different board games, and an intense game of Seven Wonders going on there. And Agricola, and a rare picture of me. Look at that. Yeah. I have only a few photos. If you don't know, I guess you can get a sense of it of how few photos I had a while back. Most of my photos just come from the past 2 years. I had a reason to take photos more frequently. But a long time ago, I only have a few. Here are some get-togethers for the Thursday night group. And again, see some familiar faces. I'll point out, and here's Jonathan at his wedding, and that was fun. That's more recent. And in this photo, this was a Thanksgiving. I'll point out Amanda here. She does this fantastic Thanksgiving celebration, and she goes crazy, and she makes like a dozen pies. All these pies on the table she made, and they're all different types of pies, and she force feeds them to us every Thanksgiving. And it's a really memorable moment. And so, again, celebrating some of the friends I've made in the past years. And she also likes board games, and so here's an intense board game session. Okay. So those were all big celebrations and big joys that I've been a part of these past 5 years. I spent a lot of time for chapter 7 here. I spent a lot of time deciding whether and how to include this chapter. because it's less enjoyable as the other ones. But I think it's also important both to remember and to recount, but also to have the opportunity to share some really pleasant and some really fantastic perspectives and connections, perspectives I've gained over the past 5 years, and seen some things in people. So I won't show any pictures for this section, but I have a few more quotes. But I'll say chapter 7, a time times half a time. Title was chosen. It's this reference, a biblical reference to this period of struggle and turmoil. And then 7 also chosen for completion. And so people who know me well know that the past years has been unpleasant. That's all I'll say about that, but I include it because of the moment that it gives me to reflect and remember some really fantastic perspectives I've seen in some people. I'm reminded of times in which I've seen someone I really, really respect and someone who I think is really strong to share a struggle that they've been having, or a frustration, a burden that they've had. And the strange encouragement it is to see that, and how rare it is to see that in our modern society of not showing it, not being vulnerable. And yeah, the strange encouragement it can be to see someone you respect and someone you think is strong to bear their burden with tenacity and hope. And so I'm reminded of a few moments in which I've seen him and her bear those burdens, and those are moments worth celebrating and worth sharing. I'm also reminded of some perspectives where I've seen people who I also respect and who I think are really well thought out in who they are and their perspectives on the world, and the beauty it is to see the human experience and how varied it is, the kaleidoscope and the different colors people have. To see someone who I think is well thought out express frustration or disagreement or struggle with something within themselves or within society that I wouldn't think twice about. That has never bothered me. And I think of those moments and the beauty it is to see the different colors of the human experience expressed, and to see the different colors that you wouldn't have been able to see by yourself shown in different people. Someone else, and finally I'll share, a friend asked me recently what has been the most memorable and positive experience these past 5 years. And again, truly, if you know me and you know these past 5 years, you know that this chapter and the predecessors has been the most substantial, both in time and in depth of heart, and is worth chronicling of my time here in New Haven. So again, this is a celebration of my story, my whole story in New Haven, celebration of some of my science, celebration of the community, celebration of the company now I share it with, and share some of the things I've learned, and hopefully you guys have learned a little bit about science or about perspectives. And so I move on. I close this chapter of my time in New Haven with hope of the adventure of the next step. And so the end, and thanks for all the fish, if you get that reference. All right, I promised a tight 1 hour. I was wrong. But unless anyone has any questions, comments, let's go celebrate and have some cake, have some food, pizza, snacks. We're all going to walk down the road. If you'll just follow me, I'll lead the food party. And then also, if you haven't already, please sign my thesis book. Write a little note. It'll be here. I'll also be at the reception, just down the road. Okay. Thank you all for coming.
David, that was congratulations.